Today, we're answering the top 5 questions we get asked most about HiFi long-read sequencing. We'll cover what makes HiFi different from short reads, how it compares to other long-read methods, how it captures methylation without any extra work, what it actually costs, and how much sample you need to get started. So let's get into it. The first question almost everyone asks is, what can HiFi sequencing do that short-read methods can't? Let's start with where short reads fall short. Short read methods produce millions of tiny snippets, typically 150 to 200 bases long. They're excellent for base level precision, but they can't span long or complex regions of the genome. And that means that they miss critical information, like structural variants, repetitive elements, segmental duplications, or GC-rich regions. You could think of it like assembling a 1,000-piece puzzle, but you're only working with pieces from the corners and the edges. You could probably fill in some of it, but the full picture stays incomplete. And sometimes what you think you're seeing is pretty misleading. With HiFi sequencing, your read length follows your DNA fragment size, from 500 to 20,000 bases, with 99.9% accuracy per read. That means straightforward analysis pipelines that resolve the complex regions short reads miss. And because HiFi sequences native DNA without PCR amplification, you capture all genetic variant types and DNA methylation in the same run. With short reads, that requires multiple assays, and even then, the picture is incomplete. What methylation markers can you see? 5MC, 5HMC, and 6MA, all from the same sequencing run. And with the Fiber-seq chromatin assay, you can go even further, mapping transcription factor footprints, nucleosome positioning, variants, and methylation from single chromatin fibers. Now that is a true multi-omic picture. If you're working with RNA, you probably know that short-read RNA-seq only gives you fragments of the transcriptome. Long reads, on the other hand, let you see full-length isoforms, so you can actually follow the splice patterns and understand gene expression without guesswork. And researchers are telling us that isoforms are where transcriptomics are headed, because seeing the whole message tells you so much more than just seeing fragments. So what advantages does HiFi offer over other long-read methods? There's no surprise here. The biggest difference is with accuracy. Because with HiFi sequencing, there's no need to average out noisy signals the way that nanopore-based methods do. Each HiFi read is highly accurate on its own. And that means no GC bias, no repetitive region dropouts, reliable performance across many different species, even the challenging ones. Benchmarking studies back this up. HiFi sequencing consistently delivers higher per-base accuracy, cleaner variant calls, and fewer false positives than nanopore-based methods. Then there's sample input. With SPRQ chemistry, you only need 500 nanograms for native whole genome sequencing. And for samples where DNA is limited, the Ampli-Fi protocol requires just 1 nanogram. Now compare that to other long-read systems, which can require several micrograms for comparable workflows. On the analysis side, HiFi data files are about 20 times smaller than nanopore-based platforms. A typical HiFi genome BAM file is only about 30 to 60 gigabytes. Now compare that to over 1,300 gigabytes for nanopore-based platforms. That's not just a storage advantage. That means faster analysis and simpler interpretation downstream. Since HiFi reads are that accurate, you only need around 20x coverage to call variants and methylation. Other long-read technologies require around 40x coverage to reach similar performance. That means more sequencing time, higher costs, and more compute. When you add in PacBio's customer and technical support, you can see why researchers choose HiFi sequencing for the most complete long-read data available. Earlier, we mentioned that HiFi sequencing captures DNA methylation automatically. So how does that work? And how is it different from other methods? HiFi detects methylation in real time, directly from the native DNA. There's no chemical conversion, no extra prep, and no additional cost. It is built into every single run. And here's how that works. HiFi measures subtle differences in polymerase kinetics. That's essentially the speed of the enzyme when it encounters base modifications like 5-MC. And that's what enables 5-base multiomic sequencing, directly linking genetic variation with methylation changes, all from the same assay. If you compare that to short-read platforms, you know that they require separate assays, extra prep, extra cost, longer turnaround time. And whole-genome bisulfite sequencing uses really harsh chemicals that can damage the DNA and distort results. Microarrays only capture a fraction of CpG sites. So you're ending up doing a lot more work and spending more money, but for less complete epigenetic information. With HiFi sequencing, you get nucleotide-level sequencing and clean, direct methylation data all baked into the same run. And with the Fiber-seq assay, you can see how methylation and chromatin structure interact at the single molecule level. The result is a complete, haplotype-resolved genome, transcriptome, methylome, and chromatin epigenome all in one go. At this point, you might be thinking, all right, this sounds great, but can I even afford it? How much does HiFi sequencing actually cost? Within the last few years, the cost gap has closed dramatically. And now, with the Vega benchtop system, high-accuracy long reads are accessible to every lab. But let's walk through some numbers. For human whole genome sequencing at 20x coverage, using the Revio system with SPRQ-Nx chemistry, you're looking at around $300 per genome with as little as 500 nanograms of sample input for prep. That's comparable to short-read input amounts, but it's the most economical long-read genome available. And for transcriptomics, you could plan for a few $100s per sample. We know the Kinnex RNA workflow costs more than basic short-read RNA-seq. But the thing is that you're not just measuring gene expression here. You're getting full-length, isoform-resolved data that reveals novel transcripts, fusion genes, and the unique UTR context. That's a fundamentally richer data set, and it's where the novel biology is being uncovered. For microbial applications, you're in the same cost range as short reads, tens of dollars per sample. But for that, you get reference-grade, closed chromosomes and plasmids, plus their methylation data. And for full-length 16S and amplicons, the per-sample cost drops to just a few dollars, with species and strain-level resolution that short reads just can't match. So with HiFi sequencing, you're not leaving any data on the table. You're capturing it all, and you're avoiding the hidden cost of rework, missing data, or any extensive bioinformatics workarounds. Ultimately, it's fewer surprises, cleaner biology, and just more from every dollar that you spend. We've covered what HiFi sequencing can do, how it compares, and what it costs. This brings us to our last question. How much DNA do you actually need? The good news is that you don't need much. Let's start with extraction. The PacBio nanobind extraction kits are fast, high yield, and gentle on long fragments, so you get ready-to-use DNA across a wide range of sample and species types. With SPRQ chemistry, you need just 500 nanograms of input DNA for a standard whole genome run. Most common extraction methods from blood, cells, or tissue will give you much more than that. So for many sample types, you're already there. For transcriptomics, you only need 300 nanograms of total RNA. And for single-cell sequencing, you can start with as little as 15 nanograms of cDNA input, compatible with many existing single-cell technologies. Now let's compare that to nanopore-based platforms, which typically need at least a microgram or even more of high molecular weight DNA. And even then, the results vary based on sample purity, fragment length, or GC content. Short reads can work with less input, but you know that you have limited insight. HiFi sequencing sits in the sweet spot. Long reads, high accuracy, and input requirements that are now in the same range. If your lab requires automation, HiFi sequencing supports automated DNA extraction and library prep on common liquid handling platforms. So what's the bottom line? High-quality, long-read sequencing is now realistic for labs of any size. There you have it, our top 5 questions asked by you. HiFi sequencing provides long, accurate reads that reveal what short reads miss. Higher accuracy data and smaller files than other long-read methods. Methylation built into every run. Costs that compete with short reads. And automation-ready workflows that let you get started faster. So, those are your top 5 questions. If you want to learn more about what HiFi sequencing can do for your research, head to PacBio.com. There you can explore example datasets and connect with our team. Thanks for watching, and we'll see you next time.